Good afternoon, everybody. I hope you guys are having a great day. In just a few seconds, I'll get we'll get everything moving along. Alondra, I have not posted these slides yet. And the reason is because I'm going to mark them up a whole lot. And so I wanted to put you know what, I'll put this draft up and then I will have a marked up version that I'll upload later. So give me just a second and I'll get this up as do you prefer PDF or PowerPoint file? Okay, cool. That will be up in just one second. Fall 1330. PowerPoint's in PDF. Say no markup. All right, I just posted that file, so if you refresh our Canvas page, you'll probably be able to access that. And it's PDF, no markup. It's on our homepage. Okay, so today is, let's see, today is October 1st. So we have, well, let's see. We've got October 1st. We've got our class meeting on Thursday, which will also be a synchronous meeting. And then we have an exam on October 8th. Now, that exam is going to be not covering a huge volume of material, but what it's going to be covering is kind of one contained thing. And what we're going to be talking about on that exam is amino acids and protein structure and protein function. So those are the three things that are all kind of encapsulated into one bubble of sorts. Um, the when it comes to protein structure and amino acids, those two things are interconnected. And the reason for that is because amino acids are the building blocks for proteins. And proteins, when they have a particular structure, they have a particular function. And what I mean by that is proteins are molecules, they are biological macromolecules. And that term alone, that's kind of a mouthful there. But what a protein looks like in three dimensions in 3D space, I always kind of get a piece of paper. I've got an index card near me and I crumple it up. And I say, oh, well, this is a protein. Notice how it's got a shape kind of on the x-axis, the y-axis, and it's got depth to it. So this is a protein. And whenever we think about a protein as having a particular shape, and you wouldn't really be able to describe this as a square or a rectangle, but it's fair to describe this as a globular protein. So it's kind of got a amorphous shape. Now, whenever I talk about the relationship between a protein structure and its function, what that means is, and I'm going to try to recreate this as best I can. We've got two proteins, and I did my best to replicate the structure. If this protein, and so just imagine that these two proteins have basically the exact same structure. If this one carries out a particular function, if this one, you know, if this one is involved in breakdown of sugars, and this one has a similar structure, chances are it also has a role in the breakdown of sugars. So there's this kind of interconnection between protein structure and protein function. So what we're getting into today is we're getting into protein structure and what are the kind of levels and the organizations of protein structure. So protein structure is characterized at its lowest level and referred to as the primary structure. The primary structure of amino acids. The primary structure of a protein is simply the sequence of amino acids from the N terminal to the C terminal. These chains may also be referred to as polypeptides. Now, let's rewind for a minute and think about last week. Last week, what we talked about were amino acids, which I'm going to label them as AA. Now, amino acids, we had one-letter codes, three-letter codes, and then they had unique shapes and that differentiated them from one another. Now, whenever I draw or think about an amino acid, I always think about them as being drawn from the N to C direction. So if you remember the backbone of all of our amino acids, they looked like this. They all had that NCC. And then on our N, we had three hydrogens and a positive charge. Then that carbon had a hydrogen. And then here we had a different group. For example, one of them might be this, just the hydrogen. This is what made different amino acids different from one another. Now, if I draw a molecule that's two amino acids put together, it's what's known as a dipeptide, and it looks a little bit like this. So I've got NCC, that's one amino acid, and then NCC, that's my next amino acid. This right here, and this right here, here's my H, and then I had a carboxyl group on the C termini. So it's an amine, which I'm going to label this as amine to carboxyl. So an amine to a carboxyl group. Now, this is what a dipeptide looks like. And so if you think about a protein, can anyone name a protein that they think of, or that whenever you hear protein, personally, whenever I hear the phrase protein or whenever I hear the word protein, the first thing that comes to my mind is steak. And steak has lots of proteins, absolutely. Um, can anyone name a protein that maybe they've purchased recently? And just a little bit of a lead there. Okay, I've got one person who said, I think of tuna. Absolutely. Tuna has lots of protein. What about if you had like a powdered protein that maybe you'd add water and put in a bottle and shake up? Does that ring a bell for anyone?
Yeah, like protein powder.
Exactly, like whey protein powder. Perfect. Now, whey protein. is actually a mixture of a couple different proteins. And we won't get into the nuts and bolts of those individual proteins, but whey is, or one of the proteins of whey is basically like, I think it's somewhere on the order of like 180 to 200 amino acids. So this would be a polypeptide, 120 to or 180 to about 200 AAs or amino acids. So it's basically 100 times this molecule right here. So it's this thing over and over and over and over again to make it, 180 to 200 amino acids long. So the primary structure is just going to be looking at it in the sequence of the amino acids. Now, an example, the primary structure of this angiotensin 2 is here, we're looking at the three-letter codes ASP, ARG, VAL, TYR, ILE, HIS, PRO, PHE. Now we could translate that to the full names, which would be aspartic acid, arginine. I'm going to check them off as I go through. Aspartic acid, arginine, valine, tyrosine, isoleucine, histidine, proline, phenylalanine. Now that what I just did would be a great example for a test question. Translate this polypeptide from its three-letter codes to its full name. Another direction. So just an example, I'm going to say EX example exam question. Go from this to say full name. Another good example exam question would be to go from that to one letter code. And to do this, does anyone remember their one letter codes or know their one letter codes? I'll get you started with the first one and then see if you can add in the remainder of them. And I'll also go with the last one. D space, space, space. What's arginine? What's the single letter code for arginine? R, perfect. Valine. V, perfect. What's after valine? There. What's tyrosines? Y, perfect. Then isoleucine, histidine, and proline. I, awesome. H, perfect. Well done. So these are both good example exam questions. And then to, basically to go in the reverse direction. Now, we we talked about how the single letter codes for the most part, or sorry, the three-letter codes for the most part are the first three letters of the amino acids. There's a handful where it doesn't really fit. The single letter codes are a little bit all over the place just because you've got some copies of them. But essentially, this right here, all of this is super powerful and a very important way of like communicating within biochemistry. And to be able to say a peptide that is, um, you know, DRIL, whatever, that's powerful because then you can identify a particular peptide and say, oh, okay, well, it's this sequence that is significant. I don't know if anyone's ever heard of any sort of, it's not, I don't know that it's necessarily hugely mainstream, but I think in some circles, like in powerlifting and bodybuilding circles, people are taking peptides. And these peptides are supposed to like increase muscle mass and make it easier to lift, whatever. Um, Many of these peptides, they have something that's like, they usually have these short codes that are something like BNP157 or something like that. And what's kind of interesting, or actually, I think I'll go with DNP. What's kind of interesting about them is if you know that I guess, kind of labeled name, it gives you some information about it. For example, I know that I looked one up that was something like this. It was like DNP 157. Well, does anyone want to guess how many amino acids long it was? You're right. It was about 157 amino acids long. Then the second part of it was this DNP. Well, it was actually just a couple of amino acids repeated over and over again. So, these have some what's the word? Anabolic effect on muscle building. So it's important to know this because it's a useful way of communicating and it's much faster than saying the entire name. Now, our next level of protein structure is known as our secondary structure. So this is the great thing that I love about biology and biologists is it's almost like they got together and were like, listen, creativity is not my strong suit. The primary or like the lowest level, we'll call it the primary structure. Okay, what about the next level? Well, we should probably call it the secondary structure. Why? It makes the most sense. It's easiest. So our secondary structure for any and every polypeptide is the way the chain of amino acids folds and coils as a result of hydrogen bonding of the backbone amide groups. Okay, so what this means is if we look back at, let's look at this structure at the top here. Yeah, this structure right here, this group and this group, they're far away from one another. But what can happen is that this whole molecule, I'm going to label it rather than it existing as like a straight line, instead what happens is this molecule will kind of curve. And whenever that curves, that means that this group right here is right here, and this group right here is right here. So these groups that were far away from one another, based on how this molecule folded, got in very close proximity to one another. And whenever they are in close proximity to one another, what they're able to do is they're able to establish hydrogen bonding. So when we talk about the secondary structure of a polypeptide or of a protein, we generally just refer to it as being a structure that's stabilized by backbone H bonds. Now, whenever I think about the backbone of a polypeptide, I go with my NCC and I repeat that NCC. Oop, sorry about that, everyone. NCC. And I identify this group right here and this group right here as the groups that can participate in hydrogen bonding. So I would definitely look out for a question like that. Which one of these groups can act or can be involved in hydrogen bonding? And oopsies. So expect a question like this. A, B, and then let's see. I'll go with an R group down here, C, and D. Which of these groups can participate in hydrogen bonding? And this would be a question that would be select all of the correct or all of the groups that can participate in hydrogen bonding, in which case your answer would be A and D. Because this group that's labeled with the lower or with the letter D, that is a backbone or that is a group that can hydrogen bond and it is a part of the backbone. Now, what happens whenever that does take place is we have two major structures. We have one known as a beta pleated sheet. Sometimes you'll hear these known as beta sheets. And this is where backbone hydrogen bonds interact. And if you imagine that a polypeptide looks like this, a straight line, or that's your primary structure of a polypeptide. Well, what's really going to happen is that molecule is going to kind of fold back on itself. And so you can have these kind of elongated structures that look like a big sheet where we have these hydrogen bonds that connect the strands of that structure of that molecule. Another or your other major secondary structure is known as the alpha helix. Now, when you think about biology or science or chemistry and you hear helix, what comes to mind? DNA. Absolutely. The DNA double helix. Now, one thing that I want to distinguish is that this is a protein and DNA is not a protein. They form similar structures, but that's just kind of the overlap there. They have that similarity, but that's kind of where it ends. So what I want you to take just as kind of a recap of what we've talked about so far is the primary structure is just the sequence of amino acids. Now I want to add to this that the type of bond that stabilizes this is found right here. I'm going to erase some of my other circles so that I'm highlighting this. I put a star by the bond that's stabilizing this molecule. This is known as a peptide bond. This is an example of a covalent bond. So many times you'll see a question, or you might see a question on an exam that asks, what type of bond stabilizes the primary structure? And the answer to that would be, well, a covalent bond stabilizes the primary structure of a protein. Your secondary structure is stabilized by hydrogen bonds, specifically backbone hydrogen bonds. So the best answer, if you were asked what type of bond stabilizes the secondary structure of a protein, it would be backbone hydrogen bonds. Now, what's important to know about this is when it comes to a polypeptide, we haven't really even talked about this R group. All of the R groups that make amino acids different from one another, that give us our way of distinguishing amino acids from one another. So if you remember, the R groups were kind of classified into a couple of different organizations. They were nonpolar, they were polar positive, polar negative, and polar uncharged. So those were our three major groups, or sorry, those were basically our four major groups. And then there was one last group, and those were our aromatic amino acids. So these groups right here, these, distinguishing an amino acid as polar or nonpolar or aromatic really hasn't become a part of the conversation when it comes to your primary and your secondary structure of a protein. We get into that when we talk about the the tertiary structure of a protein. Now, the tertiary structure of a protein, or when we talk about the tertiary structure of a protein, we're talking about this right here. This is a protein. This is folded in 3 dimensions. Okay. This is just a piece of paper, but basically, this is what a protein looks like whenever it's, completely in its functional shape. Now, the 3-dimensional shape of a protein exists due to the spatial relationships of groups that are far apart from one another on the protein chain. So one example is the protein chain of a globular protein. Now, I'll give you here, this right here is an example of a protein. Down below is another protein. The one that's the lower, so this would be fasciculated. Sulin, and this would be myoglobin. Has anyone ever heard of myoglobin or a similar globin protein? And if so, which is that protein, or what is the name of that protein that you've possibly heard of before? Maybe hemoglobin? So hemoglobin's a very important protein when it comes to like, well, doing what I just did there, breathing, taking oxygen from your lungs and taking it to your peripheral tissues like your fingers and your toes and, well, every other part of your body. Now, myoglobin here, well, let's think about this. I always like to think about protein structure in kind of a hierarchy. So at the highest level, or most basic level, we've got the primary structure of a protein. Then on our next level, we have the secondary structure of a protein. And then on our third level, we have this tertiary structure. So the primary structure is stabilized by covalent bonds, peptide bonds. Your secondary structure is stabilized by backbone hydrogen bonds. Your tertiary structure is stabilized by what are known as R groups. Now, what I want you to think about and look at here is when you look at this whole thing, this 3D structure of myoglobin, can anyone share what they think this thing that I've circled, what do you think that is? Let me erase one of my other circles. Judging by, your primary and your secondary structure, what do you think that is? Does that look at all like a helix? Perfect. And I'm not sure if you put A short for alpha. I'm going to go ahead and say yes, you did. This is an alpha helix. So whenever I think about and whenever I look at this protein, that's an alpha helix. That's an alpha helix. Here's another. Here's another. And another. There's tons of alpha helices that make up a protein. So whenever you think about the tertiary structure or the third level structure, I want you to think about that as that's a culmination of all of the forces that make the primary and the secondary structure. But what we've got here is I'm going to clear all my writing, and I'm going to look at this top protein. If we think about this molecule as a long polypeptide or as a polypeptide, here's one end of that long polypeptide. And then it's a little bit tough. I'm going to see if I can find the other end. Yeah, that's one end. Oh, no, I can't find the other. Okay, I scratch that. I'm going to go back and look at myoglobin. Here's one end of my polypeptide. And here's the other end of my polypeptide. I'm going to label this as my N termini, and this is my C termini. Now, myoglobin is like 160 amino acids long. If we were to look at, label this as amino acid number 1, and let's just pick another random spot. Let's call this one like 75. And then I'm going to say this right here. No, I'm going to call this like amino acid number 1. Let's call it 150. Now, thinking about this molecule as a primary structure, as a straight line, as a polypeptide, here's my model. Amino acid 1, here's 75, and then here's 150. Well, I'll go a little bit farther. Here's 160. If we think about this molecule as a straight line, as like a completely linear molecule, which amino acid is amino acid number 1 closest to? Is it 75 or 150? I'm going to label this as A or B. Thinking about it as a straight line, the primary structure, is it closer to 75 or 150? Basically, if you were walking on this straight line, which would you get to first, A or B?
A.
Perfect. Okay, I agree with that. Now, when this molecule folds in three dimensions, what is it closer to? Is it closer to 75 or 150, do you think? 150. You're absolutely right. Because when this protein takes on and uses all of the space in the room, then it can fold in a really unique fashion. And what that allows for is amino acid number one, that R group can interact with amino acid number 150's R group. So we can have these interactions where AA1 reacts or interacts with AA150. Now that's significant because remember what I said earlier, there's a relationship between the shape of a protein or the shape that a protein takes on and the function that a protein takes on. So these two things are interconnected. Now, that's our third or our tertiary structure. So to sum this up, this is R group interactions. Okay. Now, next up, our fourth level of protein structure is also known as the quaternary structure. So quaternary is your fourth highest or your fourth level. This is the highest level of protein structure. Highest level of protein structure. Now, that quaternary structure is where two or more tertiary structures can interact with each other to make a quaternary structure. So many proteins exist and function in the form of a quaternary structure. And they have unique names for however many proteins interact with one another to make that. There is a dimer, a trimer, tetramer, pentamer, hexamer. A dimer has two tertiary structures. Trimer is three, tetramer is four, five is pentamer, and a hexamer is six proteins folding together. Now, a great example of this is seen right here. This is hemoglobin. Hemoglobin is so important that you need to have functional hemoglobin in order to be able to breathe and run and walk and basically exist. Now, this is a tetramer. It is four different polypeptides that have folded and then they've come together to make something bigger than themselves. This one right here is one, two, three, and four. Now, my numbering scheme is a little different than what's detailed here, but that's really not relevant. That's not super important. Now, how do these different molecules, like how do they interact with one another? And the answer to that question is these also are stabilized, or this quaternary structure is stabilized by R group interactions. Okay. Now, that's always something that's a little bit confusing because if we jump back, tertiary structure is stabilized by R group interactions. Quaternary structure is stabilized by R group interactions. They're both stabilized by R group interactions? 100%. How are they both stabilized by R group interactions? Well, they're both stabilized by that because, and what makes them distinct and different from one another is that the quaternary structure is two or more, whereas your tertiary structure is just one protein. So quaternary structure is two or more proteins kind of folding together. So coming back to my pieces of paper, tertiary structure, tertiary structure. quaternary structure, tertiary structure, tertiary structure, quaternary structure. Now, some proteins, this right here, okay, so let's take these two proteins. They might have no function if they are independent of one another, but when they come together, now they're able to carry out some particular function. Now, when it comes to proteins and how they interact with one another, there are four ways for protein chains to link up with one another. One is through hydrogen bonding. Two is ionic interactions. Three, disulfide linkages. And four, dispersion forces. Now, these all should sound a lot like your intermolecular forces. So a hydrogen bond, for example, we talked about hydrogen bonds a bit. The hydrogen bond is the secondary structures occur when hydrogen bonds are formed between amide hydrogens and carboxyl oxygens. Tertiary structures also involve hydrogen bonding between side chains of amino acids. Your ionic bonds sometimes are called salt bridges. These occur when oppositely charged side chains interact with one another. You also have disulfide linkages when two cysteine residues are oxidized and form a disulfide linkage. And then dispersion forces, this is when two nonpolar side chains interact with one another. And we're going to get into kind of examples of all of these in just a little bit. And then we're going to kind of work through looking at those different examples. Now, an important note is that these interactions can also be formed between, this is not just a protein-protein interaction, but between a drug and a target protein. So whenever, if you think back to Lipinski's 5 rules for drug design, if you remember, there was a limitation on the number of hydrogen bond donors and hydrogen bond acceptors. Well, you don't want your drug to have an infinite number of hydrogen bond donors and acceptors. You want to limit them because then you're able to limit the types of interactions and number of interactions that that drug can be involved in. Now, here what we have are examples of the major types of interactions that exist between side chains or amino acid side chains. We've got hydrogen bonding. We've got ionic bonds or salt bridges, disulfide linkages, and dispersion forces. Okay, so when we look at these different options, I want you to be thinking about placing the amino acids and saying which ones can participate in which type of interaction. So what we're going to do is we're going to kind of classify and identify amino acids that are most likely to participate in one type of interaction. Okay, so what we're going to first look at is hydrogen bonding. Okay, so I'm going to be toggling back and forth a little bit between some of the, actually, here we go. Just a second. I will be able to grab this real quick. I'm going to grab a slide from one of my previous PowerPoints and copy and paste it into here. And then we're going to look at that slide and try to pick out basically the amino acids and, well, where they're going to end up. Just one second, folks. Go copy. Okay. So we're going to first look at hydrogen bonding. And so when it comes to hydrogen bonding, what we're going to look for is, actually scratch that. We're going to look for an R group that has a hydroxide and another R group that has, as one example, a carbonyl oxygen. Okay. So let's review our amino acids and talk about them in terms of, well, what are they going to stabilize? So let's look at our, okay, when you look at your non-polar amino acids, one of the things that's important to think about, and we're talking about our tertiary and quaternary structure or levels of protein structure, we are only interested in the R groups. So I'm going to draw a circle around a couple of different R groups. Now, these are three different R groups. So essentially, whenever it comes to tertiary and quaternary structure, I care about the part that's circled and the part that I'm scribbling out. I basically don't even care about because it's not relevant to protein structure conversations. Okay, so if we think about H bonding, and we're thinking we need to find either a carbonyl carbon, which this would be a great hydrogen bond acceptor, because it's got those lone pairs of electrons. And then what we probably also want to find is a hydroxyl group, which would be great as a donor. Then one other thing that we want to think about is, actually, I just mentioned this. What makes a carbonyl oxygen a good hydrogen bond acceptor? What does it have? Maybe some of these guys over here. Lone pairs of electrons. Okay. So if that's got lone pairs of electrons, and lone pairs of electrons are basically an indicator of something that's a great hydrogen bond acceptor, do you think that this right here would be a good hydrogen bond acceptor? Yeah, absolutely. So we could also be looking for a nitrogen that either has two bonds to it as being a good candidate for a hydrogen bond acceptor. So let's narrow our focus for just a second and let's come up with the single letter codes for amino acids that have a hydroxyl group, so that are going to be good hydrogen bond donors. Will asparagine? No, it's not going to be a good hydrogen bond donor because it doesn't have a hydroxyl group. Um, could anyone add? There's two that I'm looking at right now that I think are going to be good hydrogen bond um, donors. And I'll give you a little bit of a hint. Their single letter codes are right next to each other in the alphabet. Maybe something like this guy right here, S, because that has a hydroxyl group. So serine, S and Y. Oh, Alondra, great. But looking at so we got S and Y and then another one close to either one of those in the alphabet, or potentially close to one of those in the alphabet. It's kind of it's a little bit hidden in there, but threonine right here. has a hydroxyl group. So S, T, and Y are all good hydrogen bond donors. So those three amino acids all have a hydroxyl group. Okay. So they're going to be great for hydrogen bonding. Now, which amino acids, and actually, does that wrap them up? For the most part, the only other ones that might also participate in hydrogen bonding are aspartic acid and glutamic acid. So I would go ahead and expand this to say and D and E. So S, Y, T, D, E are all good amino acids for acting as hydrogen bond donors. Now, what about a hydrogen bond acceptor? Well, one that would be a good acceptor is either going to have a carbonyl oxygen, which asparagine, glutamine both have. So that would be um, N for asparagine and Q for glutamine. Those are good candidates. Now, hydrogen bond acceptor, something with a nitrogen on it, well, again, N and Q. Okay, so those are good candidates for hydrogen bond or hydrogen bonding. Now, if we jump back for a second, okay, so basically I'm going to come up with my list for my hydrogen bonding. Uh, yeah, N, Q, T, E, D, S, Y. Those are your single letter codes for amino acids that are great for hydrogen bonding. Now, next up, let's look at ionic bonding. Okay, so when we're looking for an ionic bond, or we're looking for amino acids that participate in ionic bonding, we've got NH3 and this COO minus. That COO minus might also be COOH. And the reason is that has to do with the solution that it's in. What's the pH of that solution? So when you see COO minus, think about it also as COOH. So we'll use those two interchangeably. Now, let's jump back. So we're looking for COOH or NH3. Or another way of thinking about that is ionic, basically a positive nitrogen. So which amino acids do you see with their single, go ahead and add the single letter codes for amino acids that you see as being good candidates for ionic bonding. And I'll give you, so we've got a positive nitrogen. Lysine fits that billing. The single letter code for lysine. Does anyone remember the single letter code for lysine? I'll give you a hint. Perfect. Hey, okay. Yeah. Then another positive nitrogen. KHR, perfect. Now, which amino acids have those COOH or COO minus groups? D and E. So, for to go back to kind of continue populating this, we've got K, H, R, D, E. Okay, so those are amino acids that are most likely to participate in ionic bonding. All right. Now, the next group. sorry. Next up is going to be disulfide linkages. Okay. This, one thing that's always confusing for folks is they see that S and they think, aha, serine. Well, in this case, this S is what's a part of a disulfide bridge. So this is actually a sulfur atom. So what that means is that we need to find an amino acid that has sulfur in the R group or sulfur in the side chain. Looking at this right here, which amino acids have sulfur? I'll give you a little bit of a hint. Sulfur is yellow. Which amino acids across this whole list have sulfur in them? Go ahead and use the three-letter code for these. Cis, perfect. There's one other. That has sulfur. Perfect, great, methionine. Now, one of the things is that despite methionine having a sulfur, this one is not going to participate in disulfide bonds. Only cysteine will participate in disulfide bonds. So we'll jump back here and say cysteine will participate in disulfide bonds. Okay, so we've got our list of amino acids that will be involved in hydrogen bonding, ionic bonding, disulfide bonding. The last group are what are known as dispersion forces. Now, we've got our two dispersion forces right here. We've got this methyl group, CH3, and then we have this, let's see, 3 carbons. What's a 3 carbon? Meth, eth, butyl group. We've got 3 carbons that make up this group. Now, with that in mind, carbon and carbon, or sorry, methyl group and other methyl groups, they don't really have a strong attraction for one another. And the reason for that is this group is nonpolar. This group is as well nonpolar. So they are combined two nonpolar groups. So whenever you think about basically a dividing line between dispersion forces and everything else, one thing that's common of all of these is these are all polar amino acids. These are all polar R groups, whereas your dispersion forces are your nonpolar amino acids or your nonpolar groups. And those nonpolar groups, they basically have no, it's all, I don't really know how to describe it. It's almost like you have two magnets it's like you have two rocks and you think that they're magnets. They don't have any attraction to one another. So when you talk about nonpolar groups, they have no force that's pulling them and connecting them to one another. So when you look at this list, and then you toggle back and forth between this list, this figure on the right, and this figure right here, well, your nonpolar amino acids are all displayed on the left here. And basically the only interaction that all of these are going to participate in is going to be dispersion forces. So when it comes to coming up with, so it's almost like if you drew a Venn diagram. Let's use my hands as the parts of that Venn diagram. You have nonpolar amino acids and amino acids that participate in dispersion forces only. They're the exact same. That overlap is 100%. So if you were to take every single three or one letter code here, so let's see, we've got proline, glycine, valine, PGB, alanine, methionine, leucine, AML, MAL. And then isoleucine, tryptophan, and phenylalanine, IWF. These are all of the amino acids that are going to participate and interact through dispersion forces. Now, the significance of this is that these are all forces that stabilize parts of amino or parts of protein structures, whether they're tertiary or quaternary structures. Now, these are all very important to note. The only one detail that I want to include is that, yeah, it's highlighted here. Your dis your disulfide bonds are covalent interactions, whereas your ionic, hydrogen bonding, and dispersion forces are all non-covalent interactions. Non-covalent. Here, here, and here. Okay. So when it comes to what sort of questions are you going to be expected to know, or what sort of questions should you anticipate for your exam that's a week from today? One, you should expect basically to be able to know the single letter codes. I think what I'm going to do as a figure for your exam is I'm going to take these two graphics, the non-polar and the polar graphic, and I'm going to basically put a box over all of the names. So I'll give you these structures. I'm not going to shuffle them up or anything like that, but I'm going to give you these molecules, and I'm not going to include the names, single or three-letter codes. Now, I did, I've got a list of amino acids that I know that I asked you to know about, and those were at the end of um, our last lecture. So I have to look back at those to see specifically which ones I was talking about. Um, but I think what I'm going to do, one sort of question that you'll get or that you should expect from this is to, ask to be known about or ask about the single and three-letter codes. That's important. But then what I think is a better question regarding protein structure and amino acids is if I gave you something like this. So I might give you a hand-drawn picture of an amino acid. And then I'll give you a structure like this and ask you the question, what type of interaction is displayed in the green box here? So I'll give you two amino acids and I'll say, what type of interaction is taking place here? And I would expect you to say, well, and I would want you to be able to look at something like this and say, okay, so that's a carboxylic acid. It's one of my negative charges. And then this right here is a positive charge. Okay. Referencing back to this would be an ionic bond. That's one of the questions that I want you to be able to answer. And I think is, I'm going to give you some questions that are label this amino acid. I'll give you the structure of it and I'll say, what is this amino acid? So that's kind of a memorization sort of question. It's almost like that's a good one to make a flashcard on. But then I'll give you other questions where I'm asking you something about a chemical property. What is this molecule, or what are the interactions that are taking place here? Or, give you something like this and say, um, circle the, polar or maybe not circle, but which of these groups is the polar positive group? And I mean, that would be pretty simple. You kind of just look for the positive charge. Um, these are questions that I want you to be able to answer because what they all have to do with is the overall structure of a protein. These are all building blocks in understanding how amino acids interact with one another. Now, with that said, lots of this lecture is definitions. It's defining what stabilizes the primary structure, what stabilizes secondary structure, what stabilizes the tertiary and quaternary structure. What I think one of the things that's very important for you to know about that people regularly ask about and that I will ask about is what's the difference between the primary, secondary, tertiary, quaternary structure? For instance, um, the forces that hold together the, or which forces are stronger, the forces that hold together the primary or the secondary structure. And well, the primary structure is held together by a covalent bond, and our covalent bonds are our strongest types of bonds. So the primary structure is held together by the strongest type of interaction. The secondary is hydrogen bonds, those are fairly weak. The tertiary structure, well, those could be any one of these. The tertiary structure is stabilized by hydrogen bonding, ionic bonding, covalent interactions, and dispersion forces. Um, so those are all big questions that I want you to be able to answer. Now, with that in mind, this I would say that this is a really good index card or flashcard for you to make. Um, the single letter codes of the amino acids that participate in disulfide bonds, ionic bonds, and so on. That's a good one because I guarantee you're going to get at least one, maybe two questions about this. Okay, well, that's what I wanted to cover today. On Thursday, and I'm going to upload this marked up presentation. On Thursday, we're going to have another synchronous meeting. We're going to be talking about protein function. Um, and I'm going to make a quiz go live um, later today, and you'll have until Thursday at 11 p.m. The one that I've posted or intended to post for Thursday, this past Thursday, I pressed save, but I did not press save and publish. I apologize for that. Um, but that's, yeah, there will be one posted a little bit later today and then one on Thursday. And then I think what I'm going to do on Friday is again have office hours and just be available for any questions that you may have. And then anything else that comes up over the course of the week in terms of like prep guides or study guides, I'll post for our or I'll post them for you in advance of your exam. And that's another thing. Your learning objectives for this exam, those should be up by the end of the day tomorrow with some like good questions of like this is something to think about for this upcoming exam. If anyone has any questions, you're welcome to hang around. If not, I hope you have a wonderful day and I will see you on Thursday. Thanks for coming to class, everyone.
All right, bye. Have a good day.
Thanks, you too.